So today we'll be talking about one small topic that is left in your amino acid metabolism. That is, what is proteinuria, what is hypoproteinuria, and what is hyper gamma globulinemia. So specifically coming to proteinuria, you have a very brief picture about it. You do not need to go into detail of what they are. But proteinuria is a condition where you see excessive amount of protein being excreted into your uh, in urine. Okay, that is proteinuria. Now proteinuria can be caused due to exercise, or it can be caused due to fever. It can be caused due to nephrotic syndrome. Sometimes something like acute intestinal nephritis. If there is a problem in your nephron where it is getting damaged, or the nephron itself is damaged, or it is filtering out proteins. In such a condition, you will be able to see proteinuria. Also, there are a few drugs called NSAIDs, non-steroidal anti-inflammatory drugs. Your paracetamol, indomethacin, diclofenac, all of them fall under NSAIDs. Right? You will study about them in a little more detail next year. But NSAIDs are one class of drugs that can cause proteinuria. Sometimes some infections will also cause proteinuria, something like malaria, HIV, hepatitis B, and hepatitis D, hepatitis C, sorry, and thyrotoxicosis. All right, thyrotoxicosis is a specific complication of hyperthyroidism. We already know that. So that is about proteinuria. Then we have hyperproteinemia. Hyperproteinemia is a condition in which the amount of protein in your blood is low. All right, so specifically albumin, globulin, all of those are low. Fibrinogen. So these are caused plainly due to anorexia, mostly due to problems where you are not consuming enough number of proteins, okay, through dietary sources. That is the main cause of hyperproteinemia. You can have some other causes as well. For example, renal or liver diseases. They can cause hyperproteinemia. Nephrotic syndrome can cause that. Nephrotic syndrome is again a problem in your kidney wherein it is throwing out proteins. Okay. Hemorrhage. If you have trauma leading to hemorrhage, internal bleeding, especially, you will have decrease in the number of proteins in your blood. Obviously, poorly balanced vegetarian diet. So generally speaking, when you are consuming someone who is a vegetarian, they need to be careful of the amount of protein they are eating, because generally what tends to happen, especially in cuisine like India, you can get your protein from what you call dal, but there is only so much amount of. There are other sources as well, but you have to balance them very well so that you have an overall balanced diet, not just one particular macromolecule or one but particular macronutrient in excess. All right. So that is one cause. Then you have malnutrition, where the patient is the person is not consuming, or they're not being given enough amounts of nutrition, basically generally seen in children. Starvation, they don't have anything to eat, or sometimes fat dieting as well. So people tend to get in caught up into different types of diets. So you have liquid dieting or keto or this thing and that thing. Until and unless you're balancing your proteins properly, it can cause hypoproteinemia. That will lead to different problems altogether. You need proteins in your body and to perform several functions, like for transport, for making several compounds in your body, to what do you call, uh, for integrity of your cells, and so on and so forth. There are several uses of proteins, and it is absolutely essential that you have proteins in your body and in your blood. All right. Then the last condition will be hyper gamma globinemia. This is specifically when gamma globulins are high in number. Okay, immunoglobulins, those are gamma globulins. When we discussed about proteins in blood, I told you globulins in turn have alpha, beta, gamma. Gamma globulins will be your immunoglobulins, right, responsible for immunity. Now, hyperglobulinemia can be congenital or it can be acquired. Generally, congenital you cannot do much about it; it's a hereditary disorder. But acquired is generally seen with a few conditions. So if a patient has something like multiple myeloma, multiple myeloma is a very specific kind of cancer that occurs in your B cells. Okay, now you know that your plasma B cells are responsible for the formation of immunoglobulins. If you have a cancer, or if you have a condition that is directly disturbing the levels of your B cells or destroying your B cells, then you will obviously have a problem in this. Okay, multiple myeloma is a problem in which you have a cancer of plasma B cells. There will be excessive number of plasma B cells, hence you will get excessive number of immunoglobulins. Then you have liver disease, nephrosis, uh, some chronic infections. In all of these, you will see a spike in immunoglobulins. Chronic infections pretty much straightforward. If you have an infection, your body will respond by increasing the number of immunoglobulins that are produced for protective purposes, right? So in all of these conditions, you will see hypergammaglobulinemia. So this is about the last three topics, small topics that are present in amino acids. This will complete your amino acids. We'll get into metabolism of lipids, specifically lipids. So now when we talk about lipids, though you do not have the complete classification of lipids, so I'll show you one image. Hold on. How lipids are classified. Lipids are basically esters of fatty acids and glycerol. Okay. So one second. Yeah. So lipids are basically classified into simple lipids, compound lipids, and derived lipids. First of all, what is a lipid? Lipid is a particular molecule that you get when you ester. So you have a ester bond, RCOR bond. Okay, so you have a ester bond formed between a fatty acid and glycerol molecule. Glycerol is an alcoholic molecule, meaning if I break down a lipid, I will get fatty acid and glycerol, or if I combine a fatty acid and glycerol, I will get a lipid. Okay, now they are significant importance of lipids, like they will serve as a fuel source for your body, because they are the more one of the most highest with calorific value. Now if you try to define carbohydrates and proteins in calorific value, meaning how much amount of calories you get per gram of protein or per gram of carbohydrate, it will be around 4 to 5. Whereas when we talk about calorific value of lipids, it will be around 9.3. So you get almost double the amount of calories from fat, right? But this lipids will also be responsible for insulation in your body, because it will be present in your subcutaneous tissue. It will help in what do you call, forming an insulation layer in your body, and make sure that temperature is regulated. All right, it is also important for membrane permeability. So your cell membranes, its integrity and its permeability, they are primarily what they call heavily reliant on lipids, their structure. If you do not have lipids, your cell membranes will not have their proper integrity. Other than that, one very important compound in your body is made up of lipids, which is called cholesterol. Cholesterol is very important because from cholesterol you get several products like steroid hormones, you get prostaglandins, you get vitamin D, all of that. We'll discuss about cholesterol eventually. But lipids are responsible for the formation of cholesterol in your body. You also have the cycle of cholesterol formation, so we'll discuss about that in detail later on. Other than that, your lipids will form a protective coating around your organs, a small membrane like structure. So if you look at that, it is giving integrity to your organ and protecting it. All right. So and it will serve as a source of fatty acid. If you are not, if your body is not able to synthesize fatty acids at one point of time because of a disease or because of any condition, these lipids will serve as a source of fatty acids, meaning if fatty acids are low in your blood or in your body, lipids will be broken down to get fatty acids. All right, so that is a small introduction about lipids. Now, when we come to a broad classification of lipids, you have three main categories. Okay, you have simple lipids, you have compound lipids, and derived lipids. Though in your what do you call, in your syllabus you have fatty acids and all in detail, that is a part of derived lipids. But we'll go over slightly what are individual lipids, because some of them are rather important. Something like lipoproteins, which come under compound lipids, are very important for you to understand. Now, simple lipids are basically esters of fatty acids. They are straight up esters of fatty acids, wherein you are having something like triglyceride. Okay, so when you mix glycerol and what do you call, uh, hold on, I'll show you how you get this thing. It will be easier for you to understand that way. Where is it? Yeah, so this is how you get a triglyceride molecule. Okay, this is a fat molecule, the one of the simplest form. Okay, so you take a glycerol molecule and you add three fatty acid molecules. That is a fatty acid. Now here in that fatty acid, they are taking different alkyl groups. As you can see, the R on the first R, there is one dash. Okay, so that is read as R dash. The second one has R double dash. They are not attached. Okay, they are three different fatty acids. R3 dash or R triple dash. Okay, so you get what you call, when you mix glycerol and three fatty acids, you will get a triglyceride molecule. A triglyceride molecule is one of the simplest form of fat. Okay, so that will come under simple fats. You also have waxes. Now waxes will be those which are esterified with alcohols other than glycerol. Yahan par I am forming an ester bond. So if you can see on the right here, this part specifically. So you can see that there is a RCOR bond. Yeah, that is an ester bond. I'm not able to circle it completely, but one second. Let me do it here then. Right, see RCOOR. See this part. This is RCOR. This is another. This whole thing will be counted as a single R. Okay, meaning a different alkyl group. Uh, यहाँ से यहाँ तक completely. This part will become one R, and this is the other R. So that is an ester bond. All right, the RCOR bond is an ester bond. When you have a fat or a what do you call simple lipid that is having glycerol as its acid, that will form a triglyceride. Whereas if you have an alcohol that is other than glycerol, यहाँ पर glycerol use कर रहे हैं ना, other than glycerol, if you have generally higher molecular weight alcohols, in those conditions you will get waxes, something like what do you call, myrisyl palmitate or something like hexaconal palmitate. Those are waxes. Okay, you will get beeswax, you will get different types of waxes. Now, that is about your simple waxes. Then coming to compound waxes, one second. So coming to compound waxes specifically, compound lipids, sorry. So compound lipids will contain phospholipids, glycolipids, and lipoproteins. You will have some other types as well, but these are the more important types. Now these are esters of fatty acids with alcohol that contains additional group. So your alcohol is carrying some other group with it. That is why it is forming a compound molecule. For example, if it is a fat phospholipid molecule, it is fatty acid with glycerol that has a phosphoric acid group in it. Okay, or and a nitrogenous base that will form a phospholipid. All right, you will have several types in them: lecithin, cephalins, and so on and so forth. You will not get into complete details of that, but that is one form of compound lipid where you have a different compound molecule attached to your primary structure of fatty acid and alcohol. Okay, the alcohol will be carrying some other additional group. In case of phospholipids, it will be carrying a phosphoric acid and a nitrogenous base. Okay, your nitrogenous base being adenine, guanine, and all. All right, then you have something like glycolipid. Now in glycolipid, so in compound lipids itself, you will have phospholipids, wherein it has a phosphate molecule or phosphoric acid molecule attached to the alcohol. So fatty acid and glycerol remains the same, but to that glycerol you have a phosphoric acid and a nitrogenous base attached to it. All right, then you have glycolipids. Now, glycolipids are those compound lipids that contain a carbohydrate moiety. And so you will have a fatty acid, you will have a carbohydrate moiety, carbohydrate molecule, and you have a nitrogenous base. Okay, generally, these glycolipids are seen in your myelin sheath or in your brain. Okay, and the white matter of your brain. That is where glycolipids will be present. So the main difference between phospholipid and glycolipid is that glycolipid will have a carbohydrate molecule, whereas a phospholipid will have a phosphoric acid molecule. Right? Again, glycolipids in turn will have several types in them. Not get into the details of that. Then you have something called lipoproteins. These are rather important. Why? Because lipoproteins, now you will have four different types of lipoproteins. You will have LDL, VLDL, IDL, and HDL. Okay, so you have LDL. One second, let me type it down easier. You have LDL, VLDL, HDL, IDL. Okay, in some textbooks, you might not see what you call IDL. So these are the four types. Now, what is the importance of lipoproteins? Lipoproteins are complexes of lipids and proteins, and their main function is that think of them as a special type of protein molecule that can transport a lipid molecule. Okay, so imagine this is a lipid molecule attached to the big pink colored protein molecule. Their significance is that, especially when we talk about HDL and LDL, HDL is high density lipoprotein. This fellow's work is wherever it is finding fat molecules around your body, okay, in your blood mainly, but wherever it is finding fat molecules, it will capture those fat molecules and it will transfer that fat molecule to liver. Okay, in liver what will happens is your fat molecule will get metabolized and broken down, and it will either be used up or it will be stored in what you call adipose tissue and things like that. It will be given some particular function, whereas LDL will take fat from your liver to different parts of your body. Okay, both have their own importance, but generally speaking, the amount of LDL that a healthy person has is relatively low compared to HDL. Now, whenever this ratio reverses, in the sense that whenever LDL levels increase way too high compared to HDL, that is when people will start having problems, specifically like atherosclerosis or heart problems, where fat is getting deposited in their blood vessels and things like that. Okay, I hope you are understanding. If you're not, please let me know. Uh, you can stop me at any time, either in the chat or in what you call orally. So that is a small introduction about your lipoproteins. The specific ones that you need to remember is LDL and HDL, high density lipoprotein and low density lipoprotein. You will also have very low density. These are also called chylomicrons or intermediate density lipoproteins. They're not as significant as HDL and LDL, but they do play a role in transport. They're mainly transporters of fat or transporters of lipids. Okay, to and fro, to and from liver. So liver will be the main site of metabolism of fat, to which HDL takes different fat molecules, whereas LDL will take fat molecules away from liver to the body. Okay, to wherever it is required. Now, coming to the main topics that you have, we'll be discussing about derived lipids. Okay. Now, derived lipids are derived literally, they are called derived lipids because they are derived from simple and compound lipids upon hydrolysis. Okay. So, when you hydrolyze simple and compound lipids, you will get derived lipids. For example, we have fatty acids. Now, fatty acids, once again, yeah. Now, fatty acids, these are building blocks of lipids in the sense that if you do not have, see, whenever I break down lipid, if I take a single lipid molecule and I break it down, I will get a fatty acid and a glycerol molecule. Okay, these two are essential. So, whenever I'm talking about fatty acids, it is a building block of lipid. Without fatty acid, you will not get a lipid molecule. These are basically a carboxylic acid that is attached to a hydrocarbon group. For example, I'll show you one image. Hold on. So this is a fatty acid. You will have types in them, unsaturated and saturated fatty acids. We'll get to that. But these are basically fatty acids. When we talk about the carboxylic acid chain, so this is the carboxylic acid end, COOH end, and this will be the rest of the chain. Okay, the rest of the hydrocarbon chain. So if you can see the chain that यहाँ पर line मारे, so one second. This part only contains carbons and hydrogens with the occasional double bond in between them. Whenever you will have a double bond, that means that it is unsaturated. Saturation means you cannot add any more molecules in it. Here specifically we are talking about hydrogen. Okay, unsaturated fatty acids are those fatty acids which contain double bonds, generally double bonds. You also have triple bonds sometimes, but very rarely. Okay, those double bond will represent an unsaturated fatty acid, in the sense that I can break the double bond and I can add two hydrogens here. If I do that, I will get a saturated fatty acid. Okay, saturated fatty acids are those fatty acids in which I cannot add any more molecules. So if you can see the bottom example here, you cannot add any more hydrogens to it, or you cannot add any more molecules to it. You can only replace molecule. You have to remove a hydrogen, or you have to remove a carbon. You have to remove some molecule to replace it. You cannot add anything more. Whereas in unsaturated fatty acids, you can add things. Okay, normally the natural fatty acids that you get, they are straight chain and they are even in number, in the sense that, for example, you will get something called palmitic acid, which is 16 carbon, or stearic acid, which is 18 carbons. But sometimes you get odd chain fatty acids as well, something like propionic acid, which is 3 carbon. All right, so as I was telling about fatty acids, now each fatty acid will have a free hydroxyl group, okay, on the left end, which you saw. So let me get back to that image once again. Yeah, so each fatty acid will have a free group. On the left you can see a hydroxyl group here. So this is the hydroxyl group. Okay, that hydroxyl group will be hydrophilic, meaning that part of the molecule loves water, okay, or it is soluble in water, whereas this part of the molecule is hydrophobic. Okay, the hydrocarbon chain, it is non-polar and it will be hydrophobic. It will not get dissolved in water because of the presence of both hydrophilicity and hydrophobicity. So hydrophilicity from OH end and hydroxyl end, and hydrophobicity from the hydrocarbon end, you will get a fatty acid. So generally fatty acids are amphoteric in nature. They are both hydrophilic and hydrophobic. One end is hydrophilic, the other is hydrophobic. That is how you get an amphoteric what you call molecule. Okay, amphoteric molecules are those molecules that are partially soluble in water and they are partially insoluble in water. All right, if you can understand, please let me know if you are not understanding anything. I will explain again. Okay, then if you understood, what did you understand till now? I'll ask a few questions here and there and see. Balaji, what are lipids? If your microphone, this is a problem in your microphone, you can type it down as well. Either way, whichever is convenient to you. क्या हुआ बालाजी? What are lipids? Thought you were typing, that is why I was waiting. Not able to hear your voice. Put it in the chat. Simple definition. What? How can I make a lipid? Or what is a lipid? Lipids are proteins. Lipid is a fat molecule, ma. See, how I get lipid is अपन मेटाबॉलिज्म ऑफ लिपिड्स पढ़ रहे प्रोटीन्स हो गया if I take a fatty acid, okay, and I add it to glycerol, a particular there are crows in your background, I'm muting you, okay. uh, if I take a fatty acid and I add it to glycerol molecule, an alcoholic molecule, okay, this is an alcohol, this is a fatty acid, I will get a lipid. Okay. Now you will have several types of lipids. One special type of lipid that we were discussing about was lipoprotein. Now lipoproteins are mainly lipids, but they will have a protein molecule attached to it. It is a compound molecule. I'm just asking what is a lipid. Okay. You will have lipids without proteins as well. Mainly you will have lipids without proteins. One particular type of lipid is the one which is carried by a protein. It does not have protein as such in its structure. It is only carried by that particular protein. Okay. It is a transporter. Acha. So then who should I ask? Moise, what are phospholipids? One line definition chalta. What are phospholipids?
Enzymes is an enzymes that hydrolyzes phospholipids into fatty acid.
Nahi baba, what are phospholipids? They are a special type of lipids. How are they different from normal lipids, or what is special about phospholipid?
Hydrolyzes
Phospholipid hydrolyzes, huh? Okay, anyone else?
cat.
So
Lovely.
in which
Okay, Akansha, go ahead. Go ahead. What is a
which
phospholipid?
phosphoric
It
in
has
which
a phosphoric
phosphoric
acid.
acid and a nitrogenous base
Yes.
attached to.
See, when I'm what do you call, taking a class, for your own convenience, you can write down notes, like rough notes bhi rehta chalta. Okay, so that it is easier for you to correlate later on, even if I'm asking question, or later on if you're trying to get the bigger picture what is happening bulk. So phospholipids are basically the same esters of fatty acids and glycerol, but that glycerol molecule, so you will still have a fatty acid molecule here and glycerol. Okay, but this glycerol in turn will be attached to some other molecule. They are compound lipids. That is why they are called compound lipids. They have an additional molecule attached to glycerol. That additional molecule will be phosphoric acid. So H3PO4. Okay, so H3PO4 as such nahi rehta. Okay, one hydrogen is lost and you make a bond. Okay, this will be present along with a nitrogenous base. So it might be a purine, it might be a pyrimidine. Those are nitrogenous bases. We already know what nitrogenous bases are. I hope you know. So this is a phospholipid. Okay, then lovely. What is a glycolipid?
So glycolipids contain carbohydrate molecules,
Hmm.
which it is usually seen in brains.
Hmm. Glycoproteins are generally seen in brain. So the same way यहाँ पर जैसा phosphoric acid attached था, instead of phosphoric acid you have a carbohydrate molecule plus a nitrogenous base. So you will have purine or pyrimidine. Okay, I'm just replacing the phosphoric acid with a carbohydrate molecule. You will also have something called alcohol sphygnosin, but we'll not get into that. This is the basic definition of your glycolipid. Okay, present in your brain. It will also be seen in your myelin sheet. By the way, myelin sheet क्या है? Who is myelin
Thick
sheet?
sheet present in white brain, white layer.
किसके ऊपर रहती वो?
ग्रे मैटर, व्हाइट मैटर है।
व्हाइट मैटर नहीं, क्या मॉलिक्यूल के ऊपर प्रेजेंट रहती है वो, या कौन सा सेल के ऊपर रहती है वो? एडिपोसाइट के ऊपर रहती, हेपेटोसाइट के ऊपर रहती, किसके ऊपर रहती माइलिन शीट?
Sir, axon,
एडिपो।
एक्सॉन किसका पार्ट है?
neuro,
हाँ, वो पूछ रहा हूँ मैं खुद, किसके ऊपर रहता माइलिन शीट? न्यूरॉन के ऊपर रहता ना माइलिन शीट। यू कैन नॉट हैव माइलिन शीट ऑन योर हेपेटोसाइट्स, नो, और you cannot have myelin sheath in your epithelium. What is the function of myelin sheath? What does it do? The most important function. There are several functions, but the most important function. आप लोगों को न्यूरोलॉजी पढ़ा है, या एटलीस्ट ब्रेन एनाटॉमी एंड फिजियोलॉजी पढ़ा है?
no sir.
नहीं पढ़ा है अब
No.
तक? ओके, ठीक है। तो अभी इससे पहले कुछ भी नहीं पढ़ेगा? अच्छा ठीक है, जान दो।
इलेक्ट्रिकल इम्पल्स।
Yeah, myelin sheets, neurons that are covered with myelin sheet in them, electrical impulses will move faster. Okay, because you are basically creating a resistive sheet on top. ठीक है, ऐसा अगर न्यूरॉन है, and let's suppose they are small chinks in the neuron, you already have something called nodes of Ranvier in between, right? So if I am covering this with a myelin sheet, so one second, if I cover this with a myelin sheet like this, the rate of conductance will increase, the speed of conductance will increase. Okay, that is the most important function of your myelin sheet. Anyway, you will study a little bit more about that in anatomy physiology. Coming back to our biochemistry, now as I was telling you, your fatty acids, they are the building blocks of lipids, and you will generally have a fatty acid which is made up of a carboxylic acid and a hydrocarbon chain, where one side you will have a carboxylic acid, COH, and the other side you will have a long chain of what you call hydrocarbons. Now, what is a unsaturated fatty acid? How should I ask? इसका microphone काम कर रहा, Srilakshmi, what is unsaturated fatty acid?
They can attach another groups which has to be attached. They contain double bond or triple bond.
Meaning, whenever I have a double bond, I can break this bond in between, and I can have hydrogens being attached here.
Attached molecule can attach.
Okay. So saturated molecules, saturated fatty acids, I cannot attach anything, right? All of them have single bonds.
Yes sir.
Okay, correct. Okay, we'll continue from fatty acids tomorrow. You have like 6 minutes left. Take your attendance. I hope you understand.